let us discuss about the structure of polynucleotide chain. First, here in DNA is deoxyribonucleic acid. It is a long polymer of deoxyribonucleotides. The length of DNA is usually about the number of nucleotides that are present in it. This is also a characteristic of an organism. For example, in human DNA, they consisting of 3.3 into 10 to the power of 9 base pairs. Now let us discuss about the structure of polynucleotide chain. It is a long polymer. Here you can see. It consists of the chemical nature of the polynucleotide chain, that is DNA or RNA. A nucleotide has three components. It consisting of a nitrogenous base and pentose sugar, that is ribose in case of RNA and deoxyribose in case of DNA. Okay, and a phosphate group. Here it is. There are two types of nitrogenous bases, that is purines and pyrimidines. Purines, that is adenine and guanine. Pyrimidine, that is cytosine, uracil and thymine. Cytosine is common for both DNA and RNA, whereas thymine is present in DNA, whereas in RNA uracil is present. A nitrogenous base is linked to the pentose sugar. The nitrogenous base that is linked to the pentose sugar through an N-glycosidic linkage. Okay, N-glycosidic linkage to form a nucleoside, such as adenosine. Adenine, that is adenosine, is linked together, that is, or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or thymidine, that is deoxythymidine. When a phosphate group is linked together with the OH group, a nucleoside, through a phosphate ester linkage, a corresponding nucleotide is formed. Two nucleotides are linked together to form a 3' to 5' phosphodiester linkage to form a dinucleotides chain. Then more nucleotides that are joined together in such a manner to form a polynucleotide chain. It is dinucleotide chains that is joined together in a manner, sequential manner, to form a polynucleotide chain. A polymer thus formed has one end free with the phosphate moiety at the 5' end and the ribose sugar, which is refers to as 5' polynucleotide chain. Okay. Similarly, at the end of polymer, the ribose has a free 3-OH group, that is, which refers to as 3' end of polynucleotide chain. The backbone in a polynucleotide chain is formed due to the sugar and phosphate. The nitrogenous base are linked together to form a sugar moiety project from the backbone. Here is the double helical structure of DNA. I'll explain it in later class. Okay, it's a major group. The hydrogen bonds are between them. These are the backbone of DNA, that is adenine linked with thymine with the 2 hydrogen bonds. Then guanine is linked with the cytosine with the 3 hydrogen bonds. These are polynucleotide chains where the backbone is constituted of sugar and phosphate groups and the bases project inside. These two chains will run always anti-parallel to each other, that is 5' to 3' and 3' to 5'. Okay, these are always different types of DNA is present, A, B and Z. I'll teach it in later classes. The two chains are coiled in a right-handed fashion, that is B type, and there are roughly 10 base pairs in each term, that is of 0.34 nanometer. This is a double helical structure which was proposed by Watson and Crick. They are consisting of H1 histone proteins. In eukaryotes, the organization in which more complex, there is a set of positively charged proteins that is called as a histones are present. These histones are organized to form a unit 8 molecular structure that is called as histone automer. It is called as a beads on a string. It appears like this. So it is called as beads on a string. Today class is about the structure of polynucleotide chain.